Okay, welcome everybody to a new webinar. My name is David Houteman, and I would like to introduce you today to Kaylee Ducker, our product manager MR, who will give a live demo and a presentation of all the new things in our new Medis suite MR that we are expecting in a couple of days. There will be some special focus on our new AI, a new contour detection based on deep learning, some features in 4D Flow and some other new things. Whenever you have any questions during the webinar, please feel free to use the QA section, and we'll try to answer your questions during the webinar, during the demo, or after. If there's any unanswered questions, we will make sure that we answer them by email. So this webinar will be recorded, and we'll send a link to every registrant afterwards. So with this, I would like to give the word to Kaylee Ducker for the presentation.
Thank you, David. I will start with sharing my screen. Welcome, everybody, to this webinar regarding the new upcoming release for Medis Suite 3.2. I will focus a bit on the new things that has been released previously and what are the new things that will be released in this upcoming version. So I'd like to go through the topics where we have been working on, and then I will give you a live demonstration of how it's embedded in the software. So our aim is with each release that we can provide you a better solution for your everyday cardiac MRI post-processing. And to do so, we will focus on 3 pillars to provide you with more efficiency, to provide qualitative analysis, and to provide you a robust platform. In this upcoming release, you will see that back in the following topics. So we have focused on our deep learning to get you both the LV and RV contours to get a better and efficient workflow. The qualitative analysis part is visible in the streamlines. We embedded in the 4D Flow application. So in the 4D Flow application, you can see the streamlines in there, but you can see them also in your 2D scene images. But more on that later on. And last but not least, we did implement a user logon. So this will might be required for patient data safety, bit depending on your hospital, of course, but we'll show you that. So first, regarding the LV and RV contour detection. So the first image on the left is our new algorithm, and next to it you will see our current algorithm. So to show you the difference between the two of them. This new algorithm is generated here in-house from scratch. The other algorithm was provided to us by another company. So we did a lot of work. We are very curious to your feedback regarding that. You do see in this comparison, those are the same images, and both algorithms are applied on it, that you will see that now also the right ventricle endocardial contours have been added. So this new algorithm is made in-house. It's worked on a lot of different vendors. So a lot of different vendors are in there. We will keep improving on this. So we do like your feedback. And if there are specific cases where it's not working on, please let us know so we can improve it even better. And then the next topic is the 4D Flow streamlines. So the 4D Flow application was released last October. And to get a better visualization, we added the streamlines in there. So you can see the streamlines in your 4D Flow application, but you can see it also on top of your scene images, as you can see in here. So a 2D overlay in there. I will show you a few cases in the demonstration. One very interesting one with a tetralogy. So please remind me on that. And then at last, the user logon has been added besides some other things in our platform. So with this user logon, you can just log on as you do regularly on your computer. It's the same password and so on, so you don't need to memorize an additional one. But this is really to guard for the patient data safety, that only the persons who are allowed to see the data, they can log in on this. Besides the user logon, we also did some DICOM SR outputs. We enabled that for our left and right function analysis, and it's now also possible from within the Medis suite environment to open directly our CMS monitor. But I think the main topic here is the user logon, so we'll focus on that. So in our last release, we launched our 4D-Flow application, and I hope that you can, we are very proud to present these two testimonials of our users, that they are, their feedback was very promising for us. So we will, of course, keep improving our 4D-Flow application, but I would like to show these feedback from our customers to you as well. So one is from the cardiologist at the Erasmus University Centrum here in the Netherlands, in Rotterdam, and the other one is from a medical physicist from the Helsinki University Clinic in Helsinki, Finland. Having that said, I would like to show you a demonstration of the software. So I'd like to start with the login. So for that, I will open the Medis suite, and I can log in as I would normally do. Oh, my password is incorrect. You will see that. Only have to memorize one. But after you logged in, you will see the application as it is normally. So no restrictions or anything like that, just as it appeared previously. So then the next topic, the left ventricle and right ventricle contour detection. So as you hopefully know, and otherwise please contact your salesperson, because I do think it's a really interesting feature that with our last release, we also released a method where we can automatically detect the short axis at series that comes in. So either when a new patient is loaded from your PAC scanner or from the scanner itself, that when it's loaded in or when it's pushed to the Medis suite repository, it will automatically detect if there's a short axis present, and it will automatically perform the AutoQ contour analysis on it. So you don't need to wait during. So this is an additional license is required for this. So if you haven't tried it out yet, please contact your sales representative, and then we were happy to show you how this feature is working. In the meanwhile, we can also still do it manually. I don't have a PACS or a scanner in here, so we'll show you how it manually can be done. So we select the short axis series, and then with our right mouse button, we can select a different contour detection method. So the upper one is the one that we're currently having in our release, and this is the one which is enhanced with also the right ventricle contour analysis, which will be provided in the upcoming release. So by clicking on that, the AutoQ process will start. As you might have seen in front of your patient, then you will see this working guy to show that there is something going on in the AutoQ part. In addition as well, in the current release, we provided an AutoQ status button, so you can see which of the data sets are in progress, which are already done. And so you can see also a queue if you applied it to multiple at the same time. So you can see here the process as well. I have installed it at my GPU, so it's already finished by now. You can do it either on your CPU or on your GPU, a bit depending on how you would like to have it installed. So let's go back to the browser and open this patient. so I can show you those contours. So let's go directly to the short axis, and let's load also some long axis in our QMS application. So we'll select the short axis in here. And now there are different ways to load the contours in. So I can either drag and drop them directly, and then we will see for all faces and all time slices the contours were applicable. Let's show you in a movie on how this is done. Let's zoom it a bit in so you have a bit better view on it. So here you can see over time the contouring processing. Of course, you still need to check them if there are any contours not on the right spot. But overall, I would say it's doing a good job. And the other method is to load it with the shift button pressed, and this way you will only get the contours at the end diastolic and then systolic phase. By going through, you can still apply, adapt the contours as you're used to with our software methods to make some changes if there has been some in alignment. So for example, here by freehand drawing or with the rubber banding tool, you can still change the contours in there. So based on this, you will get directly the ejection fraction from the right and left ventricle. Let's show you in another case as well. So another myocarditis case. Oh, I see there are some things already done in here. So open the QMS application. And also in here, I will drag and drop the contours onto it. Now let's focus on the end systolic and then diastolic phase. So I do see here some improvements that can be made. Overall. Well, just to give you a first impression, as said, I'm very curious to your feedback, how it's working on your cases. So please let us know. So then the last topic I would like to demonstrate to you is regarding the 4D flow dataset. So first of all, I would like to show you how it's currently looking, and then I will show you the enhancements that has been done to the streamlines. So in addition, what has also been done, we have been focusing as well to get the loading improved, since with the 4D flow datasets, we always have a lot of images. So we feel that we have made some improvements in there when it's a server client setup. But we hope, of course, that you will have the same feeling. So let us know if you do feel any difference in there. So for location-specific flow measurements, currently this 4D flow application is generated for. So the first step in there is to do a noise removal, so we can get really more focus on the flow. So it's here visualized with the speed. And I would also want to do for these measurements a background correction. So we can see the progress in here. And in the meanwhile, I can use my crosshairs to get a perpendicular view on the aorta. So you will see the result then in here, and from here we can directly launch it in our 2D flow application, where we can really do the measurements. So this 2D flow application has been in the market already for a long time. So I hope you know how it's working. So with the auto contour, with the auto button, we can get the contours in all different slices. And here in the graph, we can see our forward and backward flow and the regurgitation fractions in this case. So this is how it's how the current status is basically. And now the enhancement is that instead of the speed, we can also show you here the streamlines. So now within the 4D flow, let's make the images a bit larger so you have a better view on it. You can see the streamlines in there. So you can now see how the flow is going and where it's going to. You can still go through them. To show you that it's also possible to get the streamlines in the scene images, I will load a different case, the one I promised you, tetralogy.
In the meantime, if there's any question, feel free to ask one using the QA button that you should have in your menu.
Thank you, David. So the last one is the tetralogy case. As you can see, over 13,000 images, so those 4D flow datasets really do get big. So it's for us really a focus point to get these data loaded quickly. So I hope this is working out. Now, if you want to show your streamlines also on the long axis or short axis scene images, the easiest is to load them directly in here as well. So we'll select all of them and then go to our QFlow 4D application. So currently it's a research application, except for in the United States, there it's cleared as a clinical application. We're busy with getting it cleared also as a CE mark, but that's still pending. So in here as well, I will get rid of the noise. And I will put on the streamlines. So you can see here in the...
I think we lost Kaylee for a moment.
Yes, so I just switched back from the speed to the streamlines. I zoomed a bit in. It's taking a while to process for each time point, and then you will see all the streamlines in here. So you can still... Well, this is basically the same as I showed you in the previous case. So in our 4D flow data set, you can see the streamlines in there. In addition, what I'd like to show you here is that we also have now a T2D button. Wow. So we can also show you the images or the streamlines in the regular scene images. So in addition, I'd like to show you also the actual slice. See, I didn't load it directly. It's there already. So we can go through these slices and see the flow in there going along. Zoom in on this one. So there's a lot of flow going on, and to make sure that you only think you can focus on the real higher velocities, I can add here the 0 and change it to 20, for example. So then these streamlines you will see are only from velocities higher than 20 centimeters per second to 200 centimeters per second. So with that having done, you will see here is regurgitation of the tricuspid valve. You see we also have a crosshair in here. So if there are other images, then you will see the crosshair reference in there as well. So another nice, well, Nice. Another thing you can see here in this case is pointed out mainly in this image. So you will see, I will go over it slide by slide. It might be easier to show it to you. So here, while the heart is contracting, you will see both the flow in the pulmonary artery as in the aortic artery. And then when we go to the systole, or the end-diastolic phase, where it's relaxing again, you will see here still flow appearing in the pulmonary, or again appearing in the pulmonary artery, but you don't see it in the aortic area. So here we have a regurgitation in the pulmonary artery. I'll put it on speed so you can see it a bit. So they're both there, and now only the pulmonary artery, and then both, and then only the pulmonary artery. Very interesting that you can see it. You can see this phenomenon just because we added the streamlines in here. So if we just would show it to you like you can see it normally in a viewer, I wouldn't have seen it, but maybe you're more enhanced readers than I am. So with the streamlines, this is more easier for me to see this. Well, I do hope that these features are helping you in your clinical routine or for your research purposes. I hope you like this webinar. We are very curious on what your thoughts are on this webinar. If there are any questions, David, are there any questions currently?
So far, there are no questions. So please, if you have some questions, there's still time that we can go through. So with that, thank you very much for joining us. Yeah, hope to see you during one of the events in the future or at our next webinar. Thanks, Kaylee, for your very nice presentation.
Thank you all for joining.
Bye-bye.